Ever wondered how Frederick Sanger sequenced insulin hormone and Peher Edman refined the technique even further? In this video, I will break down the foundational principles, step-by-step methodology, flowcharts, essential chemicals, and challenges faced in these two landmark techniques. So now coming to the underlying principles of Sanger's method, this is based on N-terminal amino acid determination in different fragments of a purified polypeptide chain. This is very important to know that the polypeptide chain must be pure. If you have multiple chains in a protein, they must be individually separated and purified for sequencing. The fragment size should be between 2 to 8 amino acid residue long. Bigger size cannot be handled efficiently by this method. So the underlying principle of Edman degradation or Edman method, same thing. It is based on the sequential determination of amino acids starting from the N terminus toward the C terminus till the end in all fragments of a purified polypeptide chain. The size of the fragment should also range between 2 to 8 amino acid residue. However, this technique can handle peptide up to less than 50 amino acid residues, but it should be noted that the shorter peptide produces better result in this technique. So you have seen that in both cases short peptides are used. So how they are generated? They are generated using specific chemicals and enzymes such as trypsin, chymotrypsin or cyanogen bromide. These enzymes, for example trypsin, generate peptides which are tagged at their C terminus with arginine or lysine, whereas chymotrypsin generates fragments which are labeled with phenylalanine, tyrosine or tryptophan at their C terminus, and CNBR attack at methionine residue. So these are very helpful in aligning the final sequence. These peptides are individually separated and they are ready for sequencing. So let us go over Sanger's methodology and identify its limitations. It involves three steps. Step 1 is a reaction between dinitrofluorobenzene and short peptide at alkaline pH. It produces a derivative called DNP derivative or dinitrophenyl derivative. In step 2, this peptide is completely hydrolyzed by incubation overnight with strong HCl at high temperature. So free amino acid and DNP-AA or amino acid derivative is released, which in step 3 is identified using chromatography. There are a couple of limitations of this technique. First one is that only one amino acid is identified per fragment. You have noticed that. And also there is a complete hydrolysis of the derivative peptide. It means you cannot reuse the peptide again. Therefore, it requires large amount of protein in the range of milligram to gram quantity. However, this is best suited for small peptides and for sequencing small protein like insulin. So let us go over Edman method. So Edman degradation to contrast and compare with the Sanger's method. This also completes in 3 steps. The first step is reaction between PITC and short peptide at alkaline pH. This binds with the N-terminal amino acid, forming a derivative called PTC peptide derivative. In step 2, this is treated with mild HCl, which releases selectively the modified amino acid and intact remaining peptide with N-1 amino acid residues. In step 3, this labeled amino acid is identified using chromatography method. The intact peptide basically is reused, and this small adjustment allowed sequential decoding of protein by this method. So he used PITC, and using mild HCl, intact peptide is released, which can be reused again and again, and as a result of that, the sensitivity of the technique is high. It requires less amount of protein in the range of microgram to nanogram quantity. So Sanger method completes in 3 steps. Step 1 is reaction between DNFB and short peptide. This is known as labeling. This labeled peptide is then completely hydrolyzed in the second step using HCl, which liberate labeled amino acid plus free amino acid. The labeled amino acid is subsequently in step 3 is identified using chromatography technique. And since the peptide has been completely hydrolyzed, it cannot be reused. It means the amount of protein required increases much higher in the range of milligram to gram quantity. Second is the technique is not very sensitive, therefore the other thing is that only the first one amino acid is identified in each fragment in this method. So in summary, Edman method also complete in 3 steps. First step is labeling of N-terminal amino acid with PITC. This labeled amino acid is hydrolyzed with mild HCl, which selectively liberate the first amino acid, leaving the intact peptide, which is then recycled in the reaction. It means the amount of protein is reduced much less in the range of nanogram to microgram quantity. In step 2, the labeled amino acid is identified by chromatography, and since the peptide has been generated, the cycle is repeated again and again in each fragment to sequentially identify amino acids till the end. So the 3 steps are repeated with each remaining peptide fragment in this method. Method is sensitive and it is automated also. So finally, just to highlight Edman degradation points, it works well with shorter peptide ranging from 2 to 8 amino acid residue, although it can handle up to less than 50 amino acid residue peptide. This is most sensitive as it requires a small amount of protein in the range of nanogram to microgram quantity, and also it is easy to align and obtain the complete sequence of your target protein. This innovation of Edman has paved the way for automated protein sequencing and laid groundwork for proteomics. Just to test your memory and retention of the information from this video, there is a question: which of the following chemicals did Edman use to release the labeled N-terminal amino acid? The choices are strong HCl, mild HCl, DNFB, and the last one is PITC. The correct answer is mild HCl. So thank you very much for listening, and if you have any question, comment, please let me know. I will be happy to address your concern. And if you think it is helpful for you to understand protein biochemistry, please like it, subscribe it, and share with your friend. And thank you again. Take care. Bye bye.